Eduardo Moreno, the floor is yours. Thank you for coming.
Durval, it's a pleasure for me to be here. I'm very humble, so I have here some of my teachers when I was studying in Madrid, the Queen of Spain, the beautiful place where I'm honored to work every day. So I hope you will enjoy the talk. So, as you know, our body is composed by trillions of cells that are constantly interacting and exchanging information. But we don't feel like a trillion different parts because these cells are programmed to cooperate so that we feel like a single individual. And my interest is precisely how cells, all these many cells, form a perfect society that cooperates and makes us feel like a single individual. And we have focused on what can go wrong in social conflicts among cells. So at the beginning of my talk, I will talk basically about very basic science of how cells communicate information about their fitness. And the second half of my talk will have to deal with some observations that may be relevant for Alzheimer's disease. And essentially, I will challenge some textbook knowledge, which is always a pleasure when you have your old teachers here in the audience. So during the years we have been studying these social conflicts, what can be called generally cell competition, so cells stop cooperating and start to compete with one another, we and others have found at least 3 types of competition based on the mechanisms of this competition. The first one we discovered in Madrid in the laboratory of Ginés Morata when I was a postdoc there, and we can call it trophic competition. It's a very indirect competition where cells compete for limiting amounts of factors, for example, survival factors that are in limiting quantity, and if they don't get enough, some of the cells will die. This type of competition doesn't require necessarily cell-cell contact and doesn't necessarily lead to the selection of the best cells because cells that are closer to the source of these resources may have an advantage. The second type of competition that is based on molecules on the surface does require cell-cell contact and usually is very efficient in leading to the elimination of the less fit cells. And this is the type of competition I will be referring most of the time during my talk. So I will not explain it now, I will go into details later. And the third type of competition, which is the last one we have discovered, is more a mechanical competition for space, where cells in growing organs, when they start to over-proliferate, for example, in the cases of cancer, you can imagine that a lesion that makes cells over-proliferate will start to occupy more space, and they compress neighboring tissues, and then these cells are eliminated from the tissue, outcompeted by the over-proliferating clone or cancer cells. So the story that I'm going to tell you today started by the work of a very talented Swiss postdoc in my lab, Christa Riner, who actually has her own lab now, and we keep collaborating. And what my team and I basically discovered was that just like we can detect fitness decay in our colleagues by looking at each other, right? We can see the graying of the hair, the eye bags under the eyes, the wrinkles on the faces. We use visual cues for this, but cells can do something very similar. Of course, they don't use visual cues, but they use molecular cues. They're able to detect changes in the surface of these cells that are molecules at the membrane that are used as markers, direct markers of how healthy or how fit they are. And the crucial component of this pathway is a family of proteins called the flower proteins that are transmembrane proteins, and they can come in different isoforms. Essentially, they are very similar, but they differ in this last bit, in the last C-terminal domain. But this change completely changes the message of that these isoforms is delivering to the neighbors. So some isoforms are going to be sitting in healthy cells and tell the neighbors that this cell is very fit, is very young, and some isoforms will give the different message that this cell have a problem or is damaged, slightly damaged. So this, for example, happens during normal aging. At the beginning, there is most cells in a healthy individual will have higher levels of the UBI isoform, and as the individual ages, many more cells will start to change the profile. What has been very surprising and at the same time very rewarding for us is that many insults that in principle are very different among them, like a slower division rate, defective cell polarity, decreased metabolism, impaired endocytosis, defective signaling in several pathways like WNTs, BMPs, JAK-STAT, just cells that are totally normal but are in the wrong place in the body, in the wrong organ, slower growth, incomplete cell clusters, so cells that are not connecting with the right partners. All these things that have in common maybe just that they are creating suboptimal but viable cells. All these small defects will, through mechanisms we don't really understand, will trigger the expression of this unfit isoform, what we call the loose isoform. So to help you visualize what happened when one of these cells start to express higher levels of the loose isoforms, I will play some movies of cells in culture. So these are S2 cells from Drosophila. We can have a control here when we just mark the cells with fluorescent protein, GFP, and you see they are interacting and nothing is happening. If we now, in addition to transfecting these cells with GFP or marking these cells with GFP, we force them to express the loose isoform, so they are now going to express, the green cells are now going to express this loose isoform on the membrane. What you can see is that these cells now start to die. They explode into many different debris. They undergo cell death, and they are later on engulfed by the neighbors. You can see, again, here cells die, and they are engulfed by the neighbors. And one interesting thing in this movie is that the neighbors are very interested in what's going on with the cells. They contact them, and they're very close to them. So we ask ourselves, what happened if we do the same experiment? So now these cells is exactly the same genotype as this cell. They are GFP marked. They have the loose isoform on the membrane, but we do it at lower density, so they don't contact. So the neighboring cells do not contact this cell expressing the loose isoform, and then this cell doesn't die. So these cells need to be recognized as less fit by the neighbors in order to be killed. You can see that there are some parallelisms with the immune system, if you want, like the immune system in many occasions would do something similar. For example, when an infected cell will be expressing viral particles or bacterial epitopes, will be also recognized by the cells of the immune system and eliminated. But there are two interesting things that I want to stress that are different here. There are no specialized cells, so this happens among equal cells, so neurons with neurons, epithelial cells with epithelial cells. And the other interesting thing is that contact is not enough. And this is what I'm going to explain briefly now. So you can have the same situation in vivo. This is an epithelia where we can follow the contour of the cells. So these cells are growing. These are Drosophila epithelia. But for what matters is just any normal epithelia. You can see the contours of the cells using icadirin. Now if you, in this system, generate a situation where you, again, generate a group of cells that are less fit, in this case marked with magenta, surrounded by more fit cells, what we can see is the elimination in this growing epithelia. So we can make movies. You will see now some of these cells undergo apoptosis, and now they are extruded basally. So what Roman Levayer in my lab, a very talented postdoc, did was to calculate the probability to die of a single cell plotted against the percentage of contact that they have with the winners. Our expectation was that you need contact, so whenever you have a winner contacting the loser cell, they will die. But very surprisingly, what he found is that this was more similar to a democracy. So there was like a voting mechanism, because even if the loser cell was contacting the winner cell up to 40%, the probability to die was more or less the same. So contact was not enough. You needed to have more than 50% of the population of winners contacting the loser, and then only the probability to die start to increase until the maximum probability to die, which is when a loser cell or an unfit cell is surrounded by more winner cells. We don't know how this works, but this makes it an interesting problem to understand how this voting mechanism or this democracy of cells decides which cells should die or which cells should survive. The only thing we know is that there is an important downstream component, a new gene called Athot that computes this information and is the gene that decides whether this cell should die or not. Essentially, if you have a healthy tissue where all the cells are fit, nothing happens, Athot is not activated transcriptionally. If you have an equally unfit cell, so if all the cells are unfit and not expressing the loose isoform, again, nothing happens because there is no better cell to substitute these cells. Only if you have a group or a single unfit cell surrounded by fitter cells, then Athot is activated by this voting mechanism and triggers a cell-fitness checkpoint that gets rid of these suboptimal cells. So this is all I have to say about the basic biology, and now I will explain you what we think this may be of interest in Alzheimer's disease. So as you know, Alzheimer's disease is a devastating disease where cognitive decline is very dramatic. This is, probably you all know this, is a painter, William Utermohlen, that did self-portraits of himself since 1967 until 2000, and you can see, and he was diagnosed with Alzheimer's disease, and you can see the cognitive decline over this time. So the story I'm going to tell you is the work of Dina Coelho, a very talented Portuguese postdoc in my lab. And what we hypothesize is basically that if this is a general mechanism, and by that time we had evidence that I will not tell you today, that is the case, that the neurons also this mechanism of fitness fingerprints to select the fitter and the unfit during normal aging of the brain, for example. So we hypothesize that if Alzheimer is a gradual neurodegeneration, there is a space, there is opportunity for cells to gradually lose their fitness and start to change their fitness profile over time, so that they will reduce the expression of the healthy isoforms and start to express higher levels of the loose isoforms. So we used the Drosophila model of Alzheimer's disease, where we can overexpress the A-beta-42 protein from humans, and we characterized this model that was done by other people, not by us, but we characterized this in the brain, in the adult brain, and we could see that they had several hallmarks of Alzheimer's disease in humans, like, for example, the creation of these plaques of A-beta and the induction of cell death, of neuronal death. And then we tested our idea directly, so we looked at the flower loose isoforms, in this case marked with red. This is the adult brain of Drosophila. This is the magnification, and you can see that in a normal brain there is very little flower loose expression. This is a young animal, therefore that's what you expect. But then if you overexpress at the same age, same chronological age, you overexpress the A-beta-42 from humans in the adult Drosophila brain, you start to see that several neurons start to upregulate the loose isoform upon this insult. So then we ask ourselves, okay, is this overexpression actually eliminating some neurons from the brain of the Drosophila neuronal population? And then first we tested with tunnel. This is a marker of cell death, of neuronal death in our case. So we could test that overexpression of the human A-beta actually induces neuronal death, as you can see here in red. And if we now silence flower or Athot, what we see is a reduction in the number of neurons dying by 65% or 59%, or if you look in the mutant knockout for Athot, you can see a 66% reduction, meaning that at least in our model, most neuronal death actually goes through this pathway of flower loose and Athot expression. You can see, as expected, a correlation between the Athot expression in the dying cells. So this is a co-staining with caspases that are going to mark the dying cells and Athot, and you can see there is a correlation. And even more interestingly, it seems that this pathway is actually acting again as a fitness comparison, because if you put more loose, which one could think, okay, if I put more loose, I'm going to have more death. But this system doesn't work like that, as I already told you. If you put more loose, now all the cells have high levels of loose. Then all the cells, all the neurons think they are equally unfit, and then actually you reduce again, by 60% the death. So it's actually, again, this comparison of fitness among neurons. So to summarize this part, essentially what we think is happening during the gradual neurodegeneration of the neurons, what you have is a high percentage of neuronal death that is going through this pathway of self-fitness-based apoptosis that is eliminating the neurons that are considered by this pathway less fit. And then we became interested in this because we started reading the literature, and then we realized that there is very little experimental evidence for what is the role of neuronal death in Alzheimer's disease. So everybody assumes, we know that neurons die, right? We know that neurons die in Alzheimer's patients, and because neurons are good, we all assume neurons, if they are dying, this must be bad for you, right? But if experimentally, this has been understudied for several reasons, but one of them is that many animal models of Alzheimer's disease, if not all, actually have very little neuronal death. So it's very difficult to test functionalities. So we now had a model in Drosophila, but a nice model that would allow us to test this, and then we also had a pathway that had the majoritary death, was causing majoritary death. So we decided to test all the phenotypes or all the consequences of Alzheimer's disease in humans in the flies, like loss of memory, shortened life expectancy, impaired motor function, impaired cognitions, or the number of ventricles in the flies. And that's what I'm going to tell you now, how we tested this, the role of neuronal death in this model. This is my favorite experiment. This is a collaboration with the lab of Krista Riner and Sylvia Schwartz. So this is to test memory in flies, okay? What we do is what is called the conditioned courtship suppression assay. And this is a very funny test. So essentially, when you put a male and a female together, the male tries to courtship the female. So they sing for her by moving the wing, and they go after her singing with this movement of the wing, and then try to court her. Unfortunately, if the female is not virgin, if the female is mated, the female will reject the male. And then the male will remember this rejection. So if you take the same male and now put him with another female, even if it's a virgin female, the male is so depressed by this rejection that he will not try to court the female. So this gives you an index of the memory of the males. So we can use this nice assay, and that's what Sylvia did in Krista Riner's lab, to test what's the memory of our different genotypes. So first of all, we can have the wild type, and you see they have this memory index. The higher the memory index, the lower, the worse the memory. So if we now trigger the expression of A-beta as we would expect, and similar to, again, Alzheimer's human patients, these animals have worse memory. The memory is going bad. What happens now if we block apoptosis by removing the athot gene? So this is what happens now. Oops. I will play you the movie. This male has been rejected and is now trying to, he doesn't care. He's still trying to court the female like crazy. So essentially, he doesn't remember his previous rejection. He has forgotten about his trauma. What happens now if you do the opposite? So you can hyperactivate this pathway, and I'm not going to go into it, this is published work, but essentially if you put an extra copy of the athot gene, we know that the elimination of these loser cells is enhanced. So these cells are going to be eliminated faster, and probably you're going to eliminate even cells with not so high difference in fitness. So if we put an extra copy of athot, essentially we are hyperactivating this pathway and eliminating faster and more unfit neurons. And then what happens is that essentially these animals are very good, have very good memory of their rejection, and they again don't dare to court the female. Okay. So at least with respect to memory, it seems that neuronal death through this pathway is beneficial. So let's look at the lifespan. So we can do the same with lifespan. Essentially, this would be the wild-type lifespan, the lifespan curve of a wild type or a control fly, where we are not expressing the US A beta. If we now express the A beta 42 from humans, we can see a reduction in the lifespan of these flies, as one would expect. And then, very interestingly, if we now do an RNAi against athot, so essentially we silence athot, and therefore we block, again, more than 50% of the apoptosis, then again, this seems to be bad for the animals. Despite they have less neuronal death, they seem to be dying faster. If you look at the morphology of the brain, so you can look at the morphology of the brain. This is the brain of an animal expressing the A beta, and you can see that this brain has these vacuoles, similar again to what might happen in a human brain. What happens if we now eliminate or silence athot and the opposite, overexpress athot? Again, if we silence athot, we will see more and bigger vacuoles in the brain at the same age. As you can see here quantified, you have higher vacuoles and more. And if you overactivate this pathway by putting the extra copy of the athot gene, you slightly reduce the number of vacuoles and the size of them. So again, in the morphology of the brain, it seems that this pathway of eliminating neurons is actually beneficial. So summarizing my talk, essentially, our evidence suggests that two major messages. One, that neuronal loss occurs through this pathway of fitness fingerprints and the use of a relative self-fitness checkpoint mediated by athot. And because this pathway is eliminating the most affected cells, it seems that contrary to common knowledge, most neuronal death may have a beneficial effect and maybe a compensatory mechanism to get rid of the neurons. So essentially, we think that there is worse things than death. And there are worse things that can happen to a neuron. And it's better to kill some neurons for the function of the brain. And you may think, okay, this is just Drosophila. Should we start to think about this or not? Well, we now know that in humans, and I'm not going to talk about this much, but we now know that in humans these molecules are conserved, and they seem to have similar effects. These are, for example, work from very two talented postdocs, Isha Madan and Rajan Gonya. These are epithelial cells in culture, but if we knock out with CRISPR the flower gene, and then we transfect or infect these cells with a single isoform, you can see that in monoculture nothing happens, but you can see that if you mix those two cells, in this case, the red cells are expressing the Win isoform, and the green cells are expressing the loose isoform, you can see that in 24 hours there is a lot of selection. So it seems that the process, at least in some epithelial cells, is conserved. So we are interested actually in looking at Alzheimer patient brains now that we know what's the function of the different human isoforms. So please, if you are interested and you have access to Alzheimer patient brains, don't hesitate to contact me. And this is my final data slide. So I didn't talk about this, but we think that these different pathways may have different roles in different disease and processes. Like we know now that fitness fingerprints have a role in aging, development, regeneration, cancer, and potentially Alzheimer. And we also suspect that mechanical competition has a role during development, for example, during the migration of population of cells, and potentially also, as I also briefly mentioned, during cancer growth. So this is the persons that did the work. I already created them. As I said, this is a collaboration with the Renal Lab at the Champalimaud Foundation. And these are the founding agencies, including the Champalimaud Foundation. So thank you very much for your attention. I'm ready to take questions.
Questions? We have 2 microphones in the room.
Before they get up, can I ask Eduardo if he considers that some kind of reward is embedded on his fitness and competition mechanisms?
Some type of reward? Reward for the organism. Well, for the organism, we know it's an anti-aging mechanism. So essentially, it's a way of getting rid of cells that, if present, are going to create more trouble than benefit, right? So that's the reward. And we know that, I didn't talk about this today, but in normal aging, if you hyperactivate this pathway by putting an extra copy of ATHOT, this gene that is the cell-fitness checkpoint, essentially these animals have delayed aging, especially aging of the brain. So the brain looks better, and they live longer. They live 50% longer than a wild type, which is quite a bit.
These two mechanisms, or this mechanism with flower and azot, right?
Yeah.
When you first start showing it, we think that's a wonderful target. Then we have to remember that they exist everywhere, and they're probably part of the healthy behavior of cell competition,
Right.
and not just in disease. So
Right.
how do you go about relating this research with potential targets for therapy?
Right. So in principle, hyperactivating the pathway seems to be beneficial in general. So you are delaying aging. You can consider Alzheimer maybe just an accelerated form of aging, right? So essentially what you're doing is letting cells be more strict about the fitness of, letting animals be more strict about the fitness of the cells they keep in their body, right? So I think hyperactivating this pathway in general for all aging, one could expect, if they have a function, one could expect to have a beneficial effect. Having said that, the other problem that one can have, and I didn't talk about it today, is that cancers abuse this mechanism.
Yeah.
So it is possible, that during cancer growth, you may want to block this pathway. But I guess this is the trade-off that we have to take in many types of pathways.
Any other question? No?